uses different proteins and enzymes to break down the food particles into usable nutrients our cells can absorb. And where are the instructions to manufacture these and all the different types of proteins we need to stay alive? The instructions to make proteins are contained in our DNA. DNA contains genes. A gene is a continuous string of nucleotides containing a region that codes for an RNA molecule. This region begins with a promoter and ends in a terminator. Genes also contain regulatory sequences that can be found near the promoter or at a more distant location. For some genes, the encoded RNA is used to synthesize a protein in a process called gene expression. For these genes, expression can be divided into two processes, transcription and translation. In eukaryotic cells, transcription occurs in the nucleus, where DNA is used as a template to make messenger RNA. Then in translation, which occurs in the cytoplasm of the cell, the information contained in the messenger RNA is used to make a polypeptide. During transcription, the DNA in the gene is used as a template to make a messenger RNA strand with the help of the enzyme RNA polymerase. This process occurs in three stages: initiation, elongation, and termination. During initiation, the promoter region of the gene functions as a recognition site for RNA polymerase to bind. This is where the majority of gene expression is controlled, by either permitting or blocking access to this site by the RNA polymerase. Binding causes the DNA double helix to unwind and open. Then during elongation, the RNA polymerase slides along the template DNA strand. As the complementary bases pair up, the RNA polymerase links nucleotides to the 3' end of the growing RNA molecule. Once the RNA polymerase reaches the terminator portion of the gene, the messenger RNA,
So this is that you can figure like that. When it's actually doing the synthesis, DNA is open, and when it's go to forward, DNA again rejoin together.
Polymerase links nucleotides to...
It's like actually at the machine, here again, join DNA together, and here open the DNA and move forward.
The 3' end of the growing RNA molecule. Once the RNA polymerase reaches the terminator portion of the gene, the messenger RNA transcript is complete, and the RNA polymerase, the DNA strand, and the messenger RNA transcript dissociate from each other.
The name of this RNA known as
mRNA.
You sure?
Yeah.
It's hnRNA, first of all. Okay, sometimes it was a question. Okay, but always we select mRNA. But the first one is hnRNA, heterogeneous nuclear RNA. In eukaryotic, just not prokaryotic. In eukaryotic, and we need to remove intron and we join exon together. Finally, we can have an mRNA.
The strand of messenger RNA that is made during transcription includes regions called exons that code for a protein, and non-coding sections called introns. In order for the messenger RNA to be used in translation, the non-coding introns need to be removed, and modifications such as a 5' cap and a 3' poly A tail are added. This process is called intron splicing and is performed by a complex made up of proteins and RNA called a spliceosome. This complex removes the intron segments and joins the adjacent exons to produce a mature messenger RNA strand that can leave the nucleus through a nuclear pore and enter the cytoplasm to begin translation. How is the information in the mature messenger RNA strand translated into a protein? The nitrogenous bases are grouped into 3 letter codes called codons. The genetic code includes 64 codons. Most codons code for specific amino acids. There are 4 special codons, 1 that codes for starch and 3 that code for stop. Translation begins with the messenger RNA strand binding to the small ribosomal subunit upstream of the start codon. Each amino acid is brought to the ribosome by a specific transfer RNA molecule. The type of amino acid is determined by the anticodon sequence of the transfer RNA. Complementary base pairing occurs between the codon of the messenger RNA and the anticodon of the transfer RNA. After the initiator transfer RNA molecule binds to the start codon, the large ribosomal subunit binds to form the translation complex, and initiation is complete. In the large ribosomal subunit, there are three distinct regions called the E, P, and A sites. During elongation, individual amino acids are brought to the messenger RNA strand by a transfer RNA molecule through complementary base pairing of the codons and anticodons. Each anticodon of a transfer RNA molecule corresponds to a particular amino acid. A charged transfer RNA molecule binds to the A site, and a peptide bond forms between its amino acid and the one attached to the transfer RNA molecule at the P site. The complex slides down one codon to the right, where the now uncharged transfer RNA molecule exits from the E site, and the A site is open to accept the next transfer RNA molecule. Elongation will continue until a stop codon is reached. A release factor binds to the A site at a stop codon, and the polypeptide is released from the transfer RNA in the P site. The entire complex dissociates and then reassembled to begin the process again at initiation. The purpose of translation is to produce
But please pay attention here. We have a lot of antibiotics, which this figure and the next figure is so important for you. You should actually memorize these antibiotics. We have streptomycin. Streptomycin binds to the 30S subunit and distorts its structure, interfering with the initiation of protein synthesis. So streptomycin, by binding to 30S, doesn't allow through protein synthesis is initiation. So in addition to streptomycin, actually as you can see here, we need initiation factor 1, 3, and also IF2-GTP. IF2-GTP actually bring the forming methionine tRNA to the A site. In the next step, as you can see here, we have tetracycline. Tetracycline also interact with 30S subunit and block access of the amino acid tRNA to the A site. Actually, it's occupy the A site in 30S. Tetracycline, which is one of the most well-known antibiotics. And for elongation, we need EFTU GTP, which bring the next one to the A site. Also, we have a peptide transferase activity. Peptide transferase activity is responsible to join forming methionine to phenylalanine or next amino acid. Chloramphenicol inhibit prokaryotic peptide transferase activity. Okay? And also we have other antibiotic which is known as a promycin. Promycin actually bears a structural resemblance to amino acid tRNA and accept peptide from the P site. What is the reason? The output causing inhibition of elongation and releasing in premature termination in both prokaryotic and eukaryotic. So promycin inhibits protein synthesis in both prokaryotic and eukaryotic. By which process? By inhibition of elongation. And after that, EFGTP we need a translocation. This peptide from A site should be transferred to the P site. Okay? Translocase has this function. Erythromycin bind to the 50S and inhibit translocase activity. Okay, these other antibiotics. And all of them are repeated. Finally, releasing factor type 1, 2, and 3 help to terminate protein synthesis. They are the initiation factor, elongation, and releasing factor, and they function in both eukaryotics and prokaryotics. Again, in this table and that table, we have the function of initiation factor, which I mentioned. And the last part of translation known as a post-translation modification. After synthesis of protein, we have other steps which known as a post-translation modification. We have different kind of post-translation modification which can be triming or covalent attachment. Covalent attachment can be classified to phosphorylation, glycosylation, hydroxylation, and other covalent modifications. What does that mean? For example, in the next session, inshallah during the winter, we will study the metabolism. Gluconeogenesis, carbohydrate metabolism, protein metabolism, all of them. One of the most important activity of protein is related to phosphorylation and dephosphorylation. A lot of enzymes in our body are regulated by this mechanism. What does that mean? This mechanism acts as a key. When you add a phosphorylate or when you remove phosphorylate, it doesn't matter, it's dependent on the activity. You can lead to turn off or turn on the activity of protein by just adding the phosphorylation or removing the phosphate. It's act as a key for turning on or turning off the activity of protein. And also related to glycosylation and other And related to triming, do you know the meaning of the triming? What does that mean?
Cutting off small parts.
Yeah, exactly. When you break down, when you cleavage, what does that mean? When we need a triming, in which kind of protein? In digestive enzymes, okay? Do you have obligatory military service in your country?
Yes.
Yes, you have, exactly. Our country also have, but some country doesn't have. Why? For example, you in Pakistan?
No, I'm from India.
India, you don't have obligatory military service yet. Why some country have obligatory military service?
Because they have small population and enemies.
Not actually. For example, country like Switzerland doesn't have actually obligatory military service. Not obligatory, actually military service actually. Why we have military service?
If the people economy is better, they will not go to the military, so it's obligatory to go.
Somehow it's better to define that to be prepared against any attacks or war, fight or other thing, okay? You should be prepared. For this reason, when we are eating actually food, something, we need to digest it. If we have a digestive enzymes ready, they can also digest our stomach, our intestine cells. So we can't produce them before eating something. So it's the first one. The next one. After eating, if you are going to synthesize this protein, it takes a lot of time to transcribe their gene and then translate to the protein, and they become an enzyme to do their activity. What is the solution? The solution is triming. What does that mean? They produce these enzymes, but in pre-active form. They add a signal peptide, a protein part, to this enzyme, which this part doesn't allow them to be activated.
Until it's
we digest this part. When we eat something, we have some enzymes digest this part and they become ready and do their activity. Just when we need them, they become active. This is known as a triming.
Something like
Like key, exactly, but it's like cutting. When you cut it, it's better than the actual expression. And all of them, actually you study the enzymes. Some enzymes have a zymogen. For example, trypsinogen, chymotrypsinogen. All of them known as a zymogen. These zymogen are kind of enzymes which they need to be activated via triming. The zymogen part is removed and trypsinogen becomes trypsin, which is activated. Chymotrypsinogen becomes chymotrypsin, which is activated. Pepsinogen becomes pepsin, which also is active. All of them are like that kind of protein. So the triming, also other name is proteolysis. We have different reversible and irreversible post-translational modification. So I think is enough for protein translation. Just the next session is related to gene regulation. Just 5 minutes. Rest 5 minutes. And after that,